So the third one is spirochete, right? Spirochetes. As the name says, spirochetes are either spiral or helical bacteria. Okay, they are either spiral or helical bacteria which have flagella on both ends. Okay, that is they are lophotrichous. So how they will appear? They will appear something like this. Okay, so they have flagella on either end, so they are lophotrichous. Okay, great. What else you see? They are mainly found in aquatic environment or in free mud. Okay, free mud or aquatic environment. Okay, and a lot of these, okay, mostly they are free living, but some of these, not a lot, some of these are pathogenic. So some are pathogenic. What are they? So the pathogenic ones, they are going to cause disease in humans, and hence they are important, and they are only the question magnets. So we are going to see them. If we look at spirochetes, say for example, there is a spirochete called Treponema pallidum. Okay, so Treponema pallidum. It is obviously trinomial nomenclature, Treponema pallidum, and this particular spirochete causes a type of sexually transmitted disease, STD, in humans, which is very, very difficult to treat, called syphilis. Okay, so syphilis is a sexually transmitting disease, and it is caused by this particular bacteria, spirochete, called Treponema pallidum. Okay, now there is a cousin of this particular pathogen called Treponema pallidum pertenue. Okay, so this is called Treponema pallidum pertenue, and it causes Yaw's disease in humans. Okay, now Yaw's disease is actually a disease of skin. On this, on the surface of skin, you find these large blisters. Okay, बड़े बड़े घाव जैसे होते हैं, और वो ओपन हो जाएंगे और उनमें से ऐसे सिक्रीशन निकलेगा. ठीक है, सो बड़ा ही खराब सा दिखता है. ये एक स्किन डिजीज है, और अगेन these diseases are difficult to treat. Treponema pallidum pertenue. और क्या होता है? और हम लोग स्पाइरोकीट को अगर देखें तो से फॉर एग्जांपल leptospira. Okay, leptospira. Now leptospira is again a spirochete, and it causes a disease called leptosporiasis. Okay, leptosporiasis, which is nothing but infective jaundice. Okay, so it is infective jaundice. Great. Apart from leptospira, there is also called Borrelia. Okay, Borrelia recurrens. Now this is also a spirochete, and it causes relapsing fever. Okay, or relapsing fever. Okay, so Borrelia recurrens, that causes relapsing fever. Can you see? So these are only a few spirochetes that causes diseases in humans, that is, they are infective. Most of these spirochetes are non-infective. They live as a free living organism, either in the mud, okay, or in the water, mainly fresh water, or even in marine water. Okay, so this is spirochete. Moving on to the next, so the fourth one, let's see. So which is fourth one? The fourth one was mycoplasma, right? Mycoplasma. Now mycoplasma is an interesting organism, okay? Interesting group of organism. Why? Because these mycoplasma are again very simple monerans, and since they are monerans they will be again prokaryotic and unicellular for sure. Very simple yet highly polymorphic. Highly polymorphic. And since they are highly polymorphic, mycoplasma are many a times also known as joker. Okay, joker of the microbial world. Why we are calling it joker? We are calling it joker because just like the joker of a circus, they can change their shape, okay, very fast. Correct? So because they are notorious and they can change their shape very fast, we call them joker of the microbial world. Great. Now, one more thing that is very important here. In case of mycoplasma, cell wall is absent. Cell wall is absent. They do not have cell wall. Okay, by the way, that reminded me of a spirochete, right? Spirochete are also gram-negative bacteria. Okay, spirochete are gram-negative, just like the rickettsia. Okay, great. Now we are talking of mycoplasma. So when we talk of mycoplasma, we see that cell wall is absent. Now, if cell wall is absent, what is there? Instead of cell wall, they have a triple layer of lipopolysaccharide membrane. Okay, lipopolysaccharide membrane. Great. So the members of mycoplasma, these bacteria, do not have a cell wall. Instead, they have additional 3 layers of lipopolysaccharide membrane. Now, if it is a lipopolysaccharide membrane, it will be similar to the plasma membrane, right? So do remember. Now, because they do not have cell wall, and instead of cell wall, they have these 3 layers of lipopolysaccharide membrane, they have ability to change their shape, right? If there would have been a cell wall, it would have restricted the change in shape, correct? But they do not have cell wall. They have LPS membrane, lipopolysaccharide membrane, and hence they are quite flexible. They can change their shape. Okay, and this is why they are highly polymorphic, because of which we call them as joker of the microbial world. Okay, great. Another thing, they are very small. So if we are talking of their size, they are very, very small. Their size can range from 0.1 micrometer to about 0.5 micrometer. Okay, that is 100 nanometer to around 500 nanometer. In some cases, some mycoplasma can grow in micron range, but typically they are very small. Okay, now since they are very small and they do not have a cell wall, it is very difficult to kill them. Okay, so the mycoplasma, they are insensitive to the lysozyme, because lysozyme attacks on the cell wall and they do not have cell wall. So obviously lysozyme cannot kill them. Not only this, lysozyme and most of antibiotics. Okay, so the mycoplasma are very notorious because they cannot be killed by lysozyme and most of the antibiotics. They are very small, so very difficult to see even through compound microscope. Okay, and secondly, they are insensitive to most of the antibiotics. Therefore, the mycoplasmal contamination or infections are always difficult to treat. याद रहेगा? Great. So if we see example, what we find is there is a particular type of mycoplasma called Mycoplasma pneumoniae. Okay, now this Mycoplasma pneumoniae causes pneumonia. Okay, it causes mycoplasmal pneumonia. Now you must be knowing that pneumonia is a disorder of respiratory tract. Okay, so mycoplasmal pneumonia is caused by Mycoplasma pneumoniae, and it is also known as PPLO, that is pleuropneumonia-like organism. Pleuropneumonia-like organism, and this organism is considered as the smallest organism. Okay, this is considered as the smallest organism, PPLO or pleuropneumonia-like organism. It is a type of mycoplasma. Great. Okay, moving on to the next one, that is fifth one, right? I hope it should be fifth one. Yeah, mycoplasma was fourth. So the fifth one is actinomycetes. Actinomycetes. Now actinomycetes, always wherever there is mycetes, this suffix mycetes is used for fungi. Okay, so earlier the members of actinomycetes, the bacterial members of actinomycetes group, were considered to be fungi. Okay, and they were commonly known as ray fungi. Okay, but in reality they are not fungi, but they are bacteria. Okay, so remember it, that in reality these are bacteria, they are not fungi. They are not fungi but bacteria. याद रखेंगे आप लोग इस बात को. Now if we look at these bacteria, these are gram positive bacteria. Okay, so if they are gram positive, they would be having a thicker cell wall of single layer that is made up of peptidoglycan, right? So they are gram positive bacteria which is filamentous and branched. Okay, so if you look at this bacteria, it might look like this. This whole bacteria, right, with a nucleus like structure. Okay, so they may look like this. Now this is typical structure of fungal hyphae, and therefore earlier it was mistakenly taken as fungi, and then we called it as ray fungi. But in reality these are bacteria, a type of gram positive bacteria. Okay, now if we look at these bacteria are mainly decomposers. They are mainly decomposers. Okay, so they do saprotrophic nutrition, and they decomposes the dead and decaying matter present in the soil. Okay, and they are mostly aerobic. They are mostly aerobic. I'm not saying all are aerobic, they are mostly aerobic, or they are facultative anaerobe. Okay, so they are facultative anaerobe. Basically they are aerobic. Okay, now it is a very, very important group of bacteria because we obtain a lot of we obtain a lot of antibiotics, okay, and other pharmaceutically active compounds, okay, other pharmaceutically active compounds from this bacteria. Okay, so we actually able to get a lot of medicines, a lot of drugs from this bacteria. Okay, we are going to see some example. So if we look at example, okay, so say for example there is antibiotic called streptomycin, right? So this particular antibiotic streptomycin is obtained from a type of actinomycetes bacteria called Streptomyces graeus. Sorry. Um, okay. So Streptomyces graeus. So streptomycin is a very important antibiotic and it is obtained from Streptomyces graeus. Another antibiotic such as erythromycin, right? Now this antibiotic is obtained from again Streptomyces erythris. Okay, this is again a type of actinomycetes bacteria. Okay, so streptomyces, erythromycin, neomycin, neomycin. Okay, so neomycin is obtained from Streptomyces fradi. Okay, so again it is a what actinomycetes member. So can you see a lot of antibiotics are obtained from the actinomycetes class of bacteria, so they are very relevant and important for us. Okay, the most of these are soil bacteria, so they are obtained in the soil. So apart from the antibiotics, some members of this group, actinomycetes, are also infectious to humans and they cause disease. Okay, so what are these? You may have heard of you may have heard of this bacteria called Mycobacterium tuberculosis, right? So Mycobacterium tuberculosis causes tuberculosis, right? TB, tuberculosis, and this is a member of actinomycetes group. Not only this, Mycobacterium M. leprae, okay, leprae. Now this particular bacteria, actinomycetes bacteria, causes a disease called leprosy. ओके, जिसको पहले के ज़माने में ज़्यादा होता था, लोग हिंदी में इसे कुष्ठ बोलते थे. ओके, कुमुड़ या कुष्ठ कहते थे इसे. सो अगेन इट इज़ अ इन्फेक्शियस डिजीज and it was a disease of social stigma. ठीक है, so Mycobacterium leprae causes leprosy. Not only this, there is another bacteria called Corynebacterium diphtheria. Okay, now Corynebacterium diphtheria causes diphtheria. Okay, so can you see the members of actinomycetes are economically very important for us because they provide a lot of antibiotics and pharmaceutical compounds. At the same time, some members of this group are infectious for humans and causes diseases, okay, grave diseases such as tuberculosis, leprosy, diphtheria. Okay, so this is your what we called actinomycetes group of eubacteria. Okay, so what we have seen so far is different type of eubacteria. We earlier saw the cyanobacteria, right, which is blue-green algae or myxophyceae. Then we saw rickettsia, members of rickettsia. Then we saw mycoplasma. Then we saw actinomycetes. we also saw the spirochetes, right? So this is all about eubacteria. Now let's move to the archaebacteria. Archaebacteria. Now, when we are talking of archaebacteria, now see, this term has archae, right? Archae. Archae means primitive, okay, and bacteria. So these are primitive bacteria. These are most primitive bacteria. They are considered to be the most primitive bacteria, okay? There are still controversies, but as of now, we study that these are most primitive bacteria. Now, archaebacteria are also known as extremophiles. Remember, wherever there is word called philes, this word has come from phili, which means to love. Okay, and extreme, you know. So we call them that these archaebacteria are extremophiles. That is, they love extremes. What extremes? Extremes of which thing? They love the extreme weather conditions. Okay. So we call them extremophiles because they can thrive or they live in extreme conditions. Okay, so they live in extreme conditions. So wherever, sorry, extreme conditions. Let me correct that. So where the conditions where we think that life is not possible, such as very high salt concentration or highly acidic condition, or maybe very high temperature. Okay, so the conditions where most of the living organisms do not exist, they cannot thrive, archaebacteria flourishes there. Okay, they grow in large amount, and hence we say that they are extremophiles, that it appears as if they love the extreme conditions, and that's why in those conditions they flourish, they thrive and they flourish. Okay, so archaebacteria are also known as extremophiles. Great. Well, the archaebacteria are able to thrive in such extreme conditions because of their unique cell wall composition. Okay, so if you see the cell wall of archaebacteria, okay, so this cell wall is of course made up of peptidoglycans, okay, like rest of the monerans. However, in this peptidoglycan cell wall, there is absence of muramic acid. So muramic acid is not present in the cell wall of archaebacterial members, right? However, in other bacteria, okay, where there is a peptidoglycan cell wall, there is also muramic acid. Okay, but muramic acid is absent in archaebacteria. Apart from that, what we find is their cell membrane, okay, their cell membrane have branched lipids, okay, branched lipids, and branched lipids have a very high melting point. Okay, because of this, the cell membrane of archaebacteria remains stable even at higher temperature. Okay, whereas if you compare any other organism, okay, other than the archaebacteria and other organisms that can thrive in the high temperature, the most of the organism cell wall do not have branched lipids. They have lipids which have a lower melting point. Okay, and this melting point usually lies in between 40 to 50 degrees centigrade. Okay, so 40 to 50 degrees Celsius, and that's why these cells are only living temperature below this mark. Okay, if you increase the temperature, what will happen? The lipid bilayer will melt away, So the cell membrane will not be there, right? It will start flowing. As a result, the transport of membrane across the plasma membrane cannot be regulated. The internal environment of the cell cannot be maintained and the cell dies. Okay. But the archaebacteria or any other thermophilic organisms which are living at a higher temperature, higher than 80 degrees Celsius, they have branched lipid. Okay, not only branched lipid, they also have fatty acid having high molecular weight. Okay, so high molecular weight fatty acids. Okay, great. Now, since there is some difference, what we see is this archaebacteria has long ago deviated from the bacterial cell line or bacterial line. Okay, so this is bacteria going on, this is eubacteria, this was common ancestor, this all we will see in evolution. So there was a common ancestor. From common ancestor, this eubacteria came up, but the archaebacteria long ago deviated. Okay, it separated from the evolutionary line, and therefore what we find is the archaebacteria has a lot of difference than eubacteria, and especially it is different in terms of genetic mechanism, okay, or genetic machinery. So if you look at the genetic machinery of archaebacteria, it is more similar to the eukaryotic cells, okay, and less similar to the bacteria or eubacteria. Okay, great. What else we see? We see that since these archaebacteria are most primitive organisms and which are still present today, that's why the archaebacteria is considered as the oldest living fossil. Okay, oldest living fossil. Let me tell you, fossil is the remains of the dead, right? But living fossil are the organisms which are still present today, but without any changes for millions of years or for a very long period of time. Okay, so such organisms which are there for pretty long time and with minimum or no modifications since then, we call such organisms living fossil, right? Say for example cockroach, okay, or king crab, limulus, right? These all are living fossils. Okay, however, the archaebacteria are considered as the oldest living fossils. How old are they? Billions of years. Okay, billions of years, literally more than 2 billion years. Okay, great. So archaebacteria, if we see, archaebacteria are most primitive bacteria. They are considered as oldest living fossil. They are extremophiles and they are able to thrive in extreme conditions because of the unique cell wall and cell membrane. If you see cell wall, it is peptidoglycan without muramic acid. Cell membrane has this unique branched lipids which have very high melting point and therefore they can sustain at higher temperature. Okay, fatty acids having high molecular weight, as well as the enzymes present in archaebacteria are very robust. Okay, so the enzymes are such that they do not get denatured in these extreme conditions. Okay, they have some unique bonds. Great. So this is overview of archaebacteria. Now if we see the archaebacterial classification, we had divided archaebacteria into 3 groups on the basis of their extreme environment. Okay, now the first one was thermoacidophile, right? Thermoacidophile. What are thermoacidophiles? See, thermo means heat, acido for acid, and phile means to love, right? So the archaebacteria that thrives, okay, thrives का मतलब रहना, okay, thrives in high temperature, okay, in high temperature. And when we say high temperature, we mean more than 80 degrees Celsius and low pH, okay. When we say low pH, that is acidic condition. How much low? Less than 2, less than 2.0. Okay, so any archaebacteria that thrives in a temperature more than 80 degrees Celsius and a pH of less than 2, we consider them as thermoacidophiles, right? Where do we find them? We find them in hot springs or hot sulfur springs, okay, hot sulfur springs, or we find them in hydrothermal vents. What are hydrothermal vents? These are deep sea vents, okay, samudra ke niche. So these are deep sea vents called hydrothermal vents. Achha, apart from hydrothermal vents, we also find them as geothermal geysers, okay. Geothermal geysers are the places on earth where hot water, hot boiling water is coming out spontaneously from underground. Okay, so such places are called geothermal geysers. Not only this natural conditions, the thermoacidophiles are also found in some polluted conditions, uh, such as the acidic mine drainage. Okay, drainage. So wherever there is a mine industry, so mining is going on, the mining requires a lot of chemical treatment and most of them are acidic. So wherever this acidic mine sewage water is going, the drainage system of such mines also contain these thermoacidophilic bacteria. Okay. Now let's see some example of thermoacidophiles like thermococcus. Very easy. Thermococcus. Now thermococcus is a group of thermoacidophiles. Okay. Now one of the most notable thermococcus that we will study later in biotechnology unit in class 12 is Pyrococcus furiosus. Pyrococcus furiosus. Now this is a type of, again, thermoacidophile, and we use this bacteria to isolate its DNA polymerase. And we use this particular DNA polymerase in biotechnology for PCR, polymerase chain reaction. We will see later what these are. Okay, so a lot of enzymes of these archaebacteria are being isolated by us for the scientific or for medical or use in the medicine field. Okay, why we are so much interested? Because these enzymes are very robust. They remain active at higher temperatures. Okay, the temperatures at which we want to work at the industrial level or in the laboratories. That's why it is of prime importance for us. Great. And we do a lot. So a lot of enzymes are being obtained from this archaebacteria or other bacteria. Great. Apart from this, we also see thermophilic bacteria just like thermoplasma, okay, like thermoplasma, etc. So these are what? These are uh, thermoacidophiles. Okay. Moving to the next archaebacteria, that is halophiles. Now, halophiles. Halo means salt. If you can recall it from chemistry, in chemistry, when you were studying periodic table, there was a group called halogens, right? I think group 7A. where you have fluorine, chlorine, bromine, iodine, and these are called halogens because they react with other compounds to form salt, right? Halogens because they are going to form salt, for generation. Similarly, wherever is there halo word, HALO, it means salt. So halophiles, phile means to love. So these are the archaebacteria which love to thrive in high salt concentration. Okay, so halophiles are a group of archaebacteria that thrive in high salt concentration. Great. Now the question is how much higher concentration? We see usually 5 times higher, 5 times higher than the seawater. Okay, so the salt concentration can be as high as 5 times the seawater salt concentration, and at such a concentrated saline or salt solution, these particular group of organisms thrive, they can live. Okay, if you consider any other organism, they will immediately be plasmolized and they will die in such a high concentration, but these bacteria or these organisms flourish. Okay, so how much is this? This is around 35% of NaCl equivalent concentration. Okay, now next question would be, where do we find such place on this earth, right? We find it such as places such as Great Salt Lake in Utah, USA, right? Or you can say places like Dead Sea, okay, or Owen Lake in California. Okay, so these are some places, or many of the salt lagoons. So these are the places on earth where the salt concentration is really, really high, and that's why in these waters you find only few organisms thriving. Okay, the organisms like halophiles. Anything else cannot thrive. Remember Dead Sea, it is called Dead Sea because nothing grows in there. Why? Because the salt concentration of the sea water, the water of Dead Sea is very, very high. Okay, and only organisms like halophiles can grow in there. Great. So what are the examples of halophiles? Let us see. Sample is Halococcus. Okay, ये सब बहुत ही आसान है नाम याद करना. हेलोकोकस, कोकस इज अ टाइप ऑफ बैक्टीरिया. हेलोकोकस, राइट? या फिर हेलोबैक्टीरियम. These all are what? These all are example of halophiles. Now, few more things that I would like to add here, that these are mostly anaerobic. Okay, these are anaerobic. When we are saying anaerobic, they are obligate anaerobic. Okay, so they are totally anaerobic. If you expose them with air, oxygen rich air, they will start to die. Okay, great. Apart from this, let me tell you this, halophiles, they have pigments called bacteriochlorophyll. Okay, they have bacteriochlorophyll, which most often gives them unique purple color. Okay, and they do make their own food, so they are autotrophic. They are usually chemoautotroph, but they are autotroph. Okay, so this is about halophiles. What is next? Next and the last type of archaebacteria are called methanogens. These are called methanogens. Now see, the name says methane and gen. Gen is to generate, right? And methane, you know, is gas, methane gas, CH4. So the archaebacteria which are able to produce methane gas are called methanogens. Okay. Now, these bacteria are obligate anaerobe. They are also obligate anaerobe. So they do not thrive in aerobic condition and they produce methane gas, right? Sorry, they produce methane gas. What they do? They ferment organic compounds, okay, organic compounds into their metabolite as well as a byproduct called methane. Okay, so they produce this methane gas, and that's why the name is methanogens. Where do we find them? We find them in places like the gut of ruminating cattle. Okay, gut of ruminating cattle. Apart from this, we also find them in deep thermal vents or hydrothermal vents. Okay, submarine ecosystem, and mostly in the gut of animals, especially ruminating cattle, such as cow, buffalo, right? Uh, Cow, buffalo, and these so on. So ruminating cattle, and therefore, because of this methanogenic bacteria in the gut of cattle are able to produce this uh, methane gas, and that's why uh, when you take the cow dung or buffalo dung and put it into a gobar gas plant, these bacteria will ferment the organic matter present in the dung and produce what? Methane gas, which forms the biogas which is being utilized by us. Okay, so methanogens. Apart from this, these methanogens are also found in bogs. Bogs कहते हैं दलदल को, right? So when you go in bogs, because of water logging, there is very less aeration in the soil, so it makes a perfect anaerobic condition, and in such conditions these methanogenic bacteria thrive, where they ferment the organic food and produce methane gas. Now this methane gas bubbles out of the soil surface or the bog surface and makes the entire area filled with methane gas, and that's why most often such bogs are at a high risk of catching fire, because methane is highly inflammable gas, right? We can use it as a fuel. In fact, we use it as a fuel. Biogas, right? The main ingredient of biogas is methane, right? Okay. Now coming to the example of methanogens, archaebacteria which are methanogens, methanococcus or methanobacterium, okay, or Methiotrix. So can you see the name are quite easy and simple. So if you are being asked to write the example of archaebacteria, if it is Thermoacidophilus, write Thermococcus, Thermoplasma, Thermobacterium, right? If you are asked to write about halophiles, Halococcus, Halobacterium, methanogens, Methanococcus, Methanobacterium, Methiotrix, easier to remember. Okay, just follow this rule. Great. So this is all of the archaebacteria. Well, so what we have seen so far in the lectures is the detailed discussion of kingdom Monera. We saw the structure of eubacterium, we saw the appendages, detailed structure of the eubacterium, then we saw how they reproduce, right? And then we saw different type of eubacteria, right? We saw Gram staining, we saw the Koch postulate, so this, and then we saw the archaebacteria. So this is all about the kingdom Monera. Okay, last but not the least, I would like to tell you the growth curve of bacteria. Okay, growth curve of bacteria. So when you see a growth curve of bacteria, what you find, let me make it first. On this x-axis, we keep time. Okay, and on y-axis, we have the population density, or you can say the bacterial population, or you can say the number of bacteria. All are the same thing. Bacterial population is number of bacteria, okay, or density are the same thing. Now, initially, when you have less time, what happens? The bacterial growth is slow, but then it picks up the speed, and then it flattens down, and finally it starts to decrease. Can you see? So see up to this time, and then up to this time, and then up to this time. Can you see? So from here to here, this is first part, this is second phase, this is third phase, and this is fourth phase in the growth curve of bacterial population or bacteria. Now the first phase, can you see the rate at which the bacteria is growing, that is time versus the number. The number is increasing at a slower speed, right? So we can see that the bacterial growth is slow, and hence we call it lag phase. We call it lag phase because here the growth is not optimum, growth is slow, lag phase. Now if you see in this section, the part 2, what we find is here the rate at which bacteria is growing, or the number of bacterial cell is increasing, is very, very high. It is not linear, rather it is exponential. Okay, that's why we call it exponential phase. Okay, or we can say it logarithmic phase. Okay, or in short we can say log phase. So lag phase, and second is log phase or logarithmic phase or exponential phase. What do you find here is the speed at which bacteria is growing or they are dividing is 0. They are not effectively, overall they are not increasing, right? The population is plateau, it is remaining constant over time, and hence we call it stationary phase. This is called stationary phase. If you look at the fourth section, what you find that the bacterial population is actually what? Decreasing, and hence we call it declining phase. Okay, now so in a typical growth curve of bacteria, we find that it has 4 phases: lag phase, log phase, stationary phase, and decline phase. Okay, great. Why it happens? Let us try to understand that why it happens. Now, see, this line, this green line is a horizontal line parallel to x-axis, is actually the carrying capacity. Carrying capacity, and it is denoted by K. So it is the carrying capacity of that surrounding, of that ecosystem. Okay, so in a particular system where these bacteria are growing, it must have a carrying capacity. Say for example, in a particular place, you can keep, say for example, I am saying average classroom. In an average classroom, at max you can have 100 students. You cannot increase that number, right? If you further want to increase, the students will push each other. They won't be able to focus on their studies, right? Why? Because of lack of a particular necessary resource called space, right? Similarly, in an ecosystem, in any ecosystem, it has its own carrying capacity, and this carrying capacity is decided by the amount of food, amount of space, amount of food, space, and other factors such as light, okay, available, which is required for the growth and development of the organisms. Okay, So say for example there's a small space which has only limited food, so it can only support, say for example 10 or 20 organisms. If you further increase the number of organisms, food is not going to increase, so there will be uh, there will be competition between these organisms for the same food. The winners will get more food, but others who are losing, they will not get food, so they will start to die. So overall the size of population will what? Decrease. Okay, so this amount of food actually decide the uh, carrying capacity, that how many organisms it can carry. Well, if you see this, initially when you have very small number of bacteria in this system, okay, there were food were a lot, the carrying capacity is well beyond the number, right? So the food and other resources were not a scarcity. However, these bacteria were new in this system. They were not able to they have not yet adjusted to the system. They are not able to therefore utilize these resources at maximum speed, and that's why the rate of their growth is slow, and that's why there is a lag phase. Once they get acclimatized to this environment, now they are able to utilize the resources in this environment really quick, and they start to divide at a very high speed, at their maximum speed, that forms the log phase or exponential phase. At what speed they divide? They divide at 2 to power n. Okay, so the number will increase by a function of 2 to power n, which we have already seen, where n is the number of divisions, right? Great, and you can calculate this n by dividing the total time upon doubling time or dividing time. Great, very good. Now once the population is in log phase, the number will increase drastically, and as it approaches to the carrying capacity, now the food or other resources in the ecosystem will become limited, right? As a result, many of the members will not get sufficient food and they will die. So the bacteria are dividing at a speed, at the same time bacteria are also dying. So overall, if you calculate the net rate of growth, that will be 0. So the rate at which they are dividing, at the same rate they are dying as well. So net rate will be 0. It will appear as if the population has become stationary. It is not increasing, neither decreasing. That is called stationary phase. Now, even at this phase, what is happening? All these bacteria, while doing their metabolism, they were also adding what? Waste material, correct? They were adding waste material. So slowly over time, waste material has accumulated in this environment. Now, because of this waste material and scarcity of food, the rate at which bacterial growth or is declining will be more than their rate of growth. So what will happen? The more bacteria will die. As a result, the population size will start to decline or decrease. Getting it? So these are the 4 phases in a typical growth curve, and this growth curve, if you look at it, it looks like a S shape, right? It is S shape, and hence we call it S-shaped curve, or we call it sigmoid curve. Okay, so that's it. This is the growth curve of bacteria. So this was all that we needed to study in Kingdom Monera. Apart from this, all you need to study is you should go and read the diseases and their pathogens. Okay, so you should go and read about different bacteria and their diseases, or bacterial diseases and their pathogen. Many a times it is asked. However, it is a topic that overlaps with class 12 course. So in class 12 there is a chapter in which we study about all the diseases and their causative agents. Okay, so it is an overlapping topic we will study there as well. Here I would only advise you to go and just learn the table where you have diseases and their causative agents, especially for the bacterial disease. Great. So with this, Kingdom Monera ends here, and in next lecture we will start Kingdom Protesta. Hope you all to join in the next lecture. Till then, take care of yourself. Thank you.